Hello, my name is Kisun Pokharel, and I work as a researcher in Natural Resource Institute, Finland. I would like to thank my organizers for giving me this opportunity. And today, my presentation is about my PhD project, where we have applied RNA-seq to explore the genetic aspects of two important reproductive events that lead to ovulation and successful pregnancy in sheep. Sheep were originally domesticated in the Fertile Crescent about 11,000 to 9,000 years before present. And since then, is one of the first domesticated species, sheep have constituted an important part of global agricultural economy. And during the 20th century, due to intensive selection and intensive breeding, several modern breeds of sheep were emerged. Currently, there are much more than 1,000 breeds of sheep, which is greater than any other livestock species. And sheep are multi-purpose species. That is, they produce wool, they yield meat, and they are sometimes used as dairy animals and even as a model species for scientific research. Sheep have a varying litter size, and mainly because of that, the prolificacy has been extensively studied. And when we talk about litter size, it is typically single lamb to quadruplets in sheep. And they are typically short day breeders, which means that autumn season is preferred time for breeding. However, there are some exceptional breeds such as Finn sheep and Romanov that are capable of year-round lambing. So while higher litter size is linked to profitability, that may not always be sustainable practice. So in general, lamb mortality rate of 15 to 20% can be considered as an indicator. So in other words, in case where lamb mortality is above 15 to 20%, then higher litter size may not be desired. But if it is the other way around, it shows that there is a potential for improving the productivity. And there are two main factors that can determine the overall reproductive efficiency in sheep farming system. So the first one is the ovulation rate, and the second one is the embryo survival rate. So in addition to these two main factors, there are also other factors, including environmental factors, such as nutrition, daylight, and seasonality. And even among these external factors, for example, flushing diet has been shown to increase the estrous activity as well as ovulation rate. Throughout the active reproductive lifespan of a female, and during each reproductive cycle, a small subset of primordial follicles, pre-existing primordial follicles are recruited to the growing pool of preantral follicles where the further development occurs. And during the course of development, and especially during the final stage of follicle development, it proceeds in a web-like pattern. So as shown in the figure, and in which 3 to 4 webs lasting for about 4 to 6 days occur in case of ewes. And from these follicular webs, one or more dominant follicles will be ovulated. And especially in prolific breeds like Finn sheep, it has been shown that follicles from 2 consecutive waves can be ovulated simultaneously. And among other factors, several members of transforming growth factor beta family have been shown to have important role in follicle development. And especially the SNP mutations in some of these well-known genes such as GDF9, BMP15 are known to influence folliculogenesis and consequently the ovulation rate. And another factor uh, which is relevant to the project and also which is highly significant in sheep reproduction is early pregnancy prior to implantation. So without going much further into detail, I just want to point out that the embryo enters uterus about 4 to 6 days after mating, and by 7th day, the blastocyst formation takes place. And the blastocyst stage of embryo development, it marks the beginning of pregnancy establishment. However, a maternal recognition of pregnancy occurs only about 12 to 13 days after mating, and interferon tau, which is produced by the trophectoderm of an elongated conceptus, acts as a signal for pregnancy recognition. And implantation in sheep involves addition of the mononucleated cells of the trophectoderm to the luminal epithelia of the endometrium. And during this stage, embryo development, embryo survival, and implantation is critical for successful pregnancy. Um, the following were the main goals of the study. So we were first interested to, in general, get the transcriptome overview of key reproductive tissues during follicular growth phase, which is ovary, and also during early pregnancy prior to implantation. So for that, we sequenced corpus luteum and endometrium. And by using a highly prolific and low prolific textile sheep, we were interested to have a breed-wise gene expression differences. And by including F1 crosses of these two purebreds, we wanted to explore how the gene expression pattern occurs in the crossbred, or if there is any parent of origin effect during gene expression. And as one of the important external factors, we included a diet in the experiment to study if there is any flossing, if there is any effect of flossing diet during early reproduction. The whole project was addressed from 3 experimental settings: pilot experiment, main experiment, and an additional experiment. And due to the constraint with time, I'm only going to talk about the main experiment. And for the main experiment, we initially selected 31 ewes of fin-sheep texel and their F1. And also at the same time, we started the diet experiment by keeping half of the ewes in flossing diet. And after about 1 month, when the ewes were showing estrus, one of the ovaries was surgically removed and biopsied for RNA-seq. And after about 3 weeks, the ewes were mated by rams, and then after next 2 weeks, they were slaughtered at different time points during pre-implantation phase of the pregnancy. At the slaughterhouse, we collected additional samples, including CL and endometrium that were sequenced. So we also had additional experiment for comparing the transcriptome profiles of selected reproductive tissues between the domestic fin-sheep and feral European mouflon, but I won't be presenting those results here. And we have used well-established tools and pipelines for the data analysis and for analyzing the RNA-seq data. So now I will share some of the results from the main experiment and about the phenotype observations. On average, FinShip showed the largest number of follicles, and which was followed by Texel and F1. The number of cells and embryos indicated reproductive efficiencies of the breeds. What is interesting to note in this table is that FinShip had the lowest survival rate for embryos. So, for example, if we compare the pure breeds, 2.6 embryos on average were obtained from 4.1 CL, which is roughly 60%. So even worse was for F1 crosses, as the embryo survival rate was approximately 50%. So on the other hand, in textile sheep, embryo survival rate was close to 90%, as 1.5 embryos were obtained from 1.8 CLs. So our conclusion from this was that possibility for improving reproductive efficiency in FinShip and F1 were better than Texel. So next, some results from the first phase of the our experiment, where we sequenced ovary RNA-seq data. And one of the main observation from this data was that the gene expression profiles of F1 crosses were more closer to FinShip than Texel, and were somehow linked with the phenotype observations that I just presented. So all the differential expression analysis revealed fewer genes between FinShip and F1 than between Texel and F1. So as you can see in the figure, F1 use share about half the genome is from FinShip and Texel. So this data is from SNP genotype data. So what we conclude from this is that gene expression is um, uh, gene expression based relatedness of F1 use cannot be explained by the Mendelian inheritance, and that there might be other parent of origin effects such as genomic imprinting, which uh, make F1 more closer to FinShip. However, this certainly needs to be studied further. And another finding from this experiment was that the results from the differential expression revealed that the effects of flossing were more pronounced in Texel than F1 or even FinShip. And actually in FinShip, we did not see any effect. So there was null effect was observed in FinShip. So a total of 118 genes were differentially expressed between the flossing and control group of Texel use. And there were no differentially expressed genes between the same comparison in FinShip. Um, Interestingly, we did not observe any of the major candidate genes. They were neither highly or differentially expressed. However, when we made manual checking, the known mutations, manual checking of the known mutations, so we observed a mutation where valine in 371 position is changed to methionine. So the mutation was seen in about half of the FinShip and F1, but none in Texel. So we further evaluated the finding are by screening this mutation in a larger population using Sanger sequencing, and then it showed that the FinShip carried this mutation with a frequency of 0.7. So which thus validated our observation. And now going to the second phase of the main experiment. So as you can see in the PCA plot, the gene expression profiles of CL and endometrium were remarkably different. And they are mainly grouped into two main clusters. And here, the more condensed cluster on the left is for CL, and the one on the right is where there are for the two subclusters is for endometrium. So what we noticed here was that the two subclusters that were formed by samples uh, uh, were coming from ewes that were slaughtered 7 to 10 days after mating, and the other from 13 to 17 days after mating. So these two subclusters basically uh, reflect the two anatomical changes in the embryos and resulting physiological change in the uterus. So after day 13, the elongating embryo secretes interferon tau, which is also known as the pregnancy recognition signal. And after the secretion of this interferon tau, it results into a remarkable gene expression changes in the endometrium. Uh, by taking embryo anatomy into consideration, ewes that were slaughtered after 7 to 12 days had spherical embryos, and those slaughtered 13 to 17 days after mating had elongated embryos. So we then compare these two groups uh, in terms of gene expression, and the comparison resulted 1,410 differentially expressed genes. So as expected, we observed many interferon stimulated genes which were upregulated in the endometrium having elongated embryos. We also noticed that the gene level information for samples having spherical embryos were relatively limited, um, thus somehow reflecting maybe the knowledge available for earlier stage is rather limited. So in particular, in the endometrium samples, we observed several transcripts of endogenous retrovirus, ERVs. So these genes are copies of retroviral genomes that have been integrated into the host genome during the course of genome evolution. So previous studies have shown that these ERVs are important during pregnancy, for example, in placental morphogenesis and embryo elongation. And uh, when we were annotating these novel genes that we found in the endometrium transcriptome, we noticed that one of the novel transcript was actually associated with the endogenous retrovirus. And more than that, uh, the transcript was found to be part of a locus, which was previously known to be associated with prolificacy in French Lacaune breed. So we speculate, based on this finding, we speculate that being part of this important locus, uh, the endogenous retrovirus may also influence the overall productivity. But for this also, maybe we would need more research for validation. And we made a comparative transcriptome of CL in FinShip, TextL, and F1. And from the analysis, we identified several novel genes which were activated during early pregnancy. They were we reported for the first time, and then also genes upregulated in FinShip. They were mainly related to immune system. Uh, and such as chemokines and then some other immunoglobulins, those are mainly upregulated. And one of the candidate genes that was related with embryonic diapers was also upregulated in FinShip and F1 compared to Texel. So based on this, we speculate that maybe the embryonic diapers is possible, or they have this kind of mechanism also in sheep. And another important observation was that maybe the flossing diet has not as promising effect in during the early pregnancy as during the folliculogenesis. So from the microRNA-seq data analysis, so we have quantified more than 500 microRNAs, which represent already more than 3x the existing microRNAs available in the MIR-based database. And one of the interesting results from the microRNA data analysis was the identification and characterization of a maternally imprinted microRNA cluster in on chromosome 18. So this particular cluster is located within 40 kilobase region and is conserved in all placental mammals. And in addition to sheep, we were also able to identify this cluster in several other mammals, such as elephant, dog, goat, pig, and so on. So with 46 microRNAs, the cluster in sheep is probably the largest in any species. And the closest was in mouse, which was about 43. So with this, I'd like to present the conclusions. So we noticed that flossing was useful for low prolific Texel, and especially during folliculogenesis. And F1 crosses were more closer to Finship than to Texel based on the gene expression analysis. And the importance of immune system during early pregnancy and possibly influencing the prolificacy was indicated by our data. And the gene expression in the endometrium was influenced by the embryos, and more remarkable effect was seen after the elongation stage of the embryo development. And we have identified or we have at least predicted that a novel endogenous retrovirus in the L locus might be associated with prolificacy or reproductive efficiency. And we have also identified maybe there is a sign of embryonic diapers in sheep. And the microRNA resource that were identified in the thesis are going to be a valuable resource, and the identification of maternally imprinted chromosome 18 microRNA cluster is a new finding as well. So with this, I would like to acknowledge my supervisors, Professor Juha Kantanen from Natural Resource Institute, Finland, and Professor Menghua Li from China Agricultural University, and all my colleagues that have helped during these years. And thank you for listening.